All right, let's talk some more about some prokaryotes. What we're going to talk about here is the different modes of reproduction for prokaryotes, their metabolism and general lifestyles, how they obtain energy and what they do with that energy, and also, so go ahead and start with reproduction. There are in terms of genetic diversity, even though many prokaryotes undergo asexual reproduction, remember the difference between sexual and asexual reproduction is that sexual involves two different organisms and they're usually genetically different from the parent, or the offspring are usually genetically different from the parents. With asexual reproduction, it's like mitosis and eukaryotes that you learned in General Biology 1. You start out with one cell and you end up with two identical or genetically identical cells. Now, they're not perfectly identical in general, and there's three other factors that contribute to genetic diversity, at least in prokaryotes, right? So in general, most organisms have a generally low mutation rate. However, in prokaryotes, because of their rapid reproduction, it only takes some prokaryotes as little as 20 minutes to double. Okay. And then this results in very large populations. So, and then of course, you know, the generation time, which is part of the rapid reproduction. It's a short generation time. Like I said, they could be as little as 20 minutes. Okay. All of this together basically contributes to more genetic diversity, right? So even though the mutation rate is low, the rate has to do with per generation, right? So if the generation is a short if it's a short generation time, there's going to be more generations. So that actually increases the number of mutations, right? And which increases the genetic diversity. So binary fission is their asexual method. So basically, it's a lot simpler. It's similar to mitosis, however, it's a lot simpler, right? So you're dealing with cells that do not have a whole lot of internal components. So there's not a whole lot of things that need to be, I guess, duplicated in a way. Definitely the chromosome needs to be duplicated. So DNA replication, of course, is going to occur. So then you're going to end up with two copies of each of the chromosome, and then the cell starts to separate. Now, there's a lot of mechanisms going on in the separation of the cell. However, it's something that you do not need to know for this course. Just know that asexual reproduction is the start of one cell dividing into two cells, which are essentially genetically identical. Genetic recombination is I guess you could call it the sexual form. This is the one that results in the most variation, and it's usually done by what's called horizontal gene transfer. And you might have been introduced to it, again, in a former class or one of our former chapters. But we're going to talk about these a little bit more in detail right now. Now, there are three modes of horizontal gene transfer. There's transformation, there's transduction, and there's conjugation. So we're going to talk about each one of these a little bit more in detail. Transformation, or the process of being transformed, is basically the process in which another cell takes up DNA from the environment. Okay, so what can happen is that a prokaryotic cell might lyse, right? So let's say it dies and it breaks apart and then it releases its fragmented genome out into the environment. Well, another cell, think about this, an environment is going to be, I guess, in this case, maybe adverse, not very good. So there's all the cells are just trying to survive. And let's say one dies, leaves its DNA behind, and then the other ones can take up that DNA. And of course, that allows it to adapt to that environment, right? So different combinations of alleles, new genes being incorporated into a genome allows it to be able to adapt to that environment a little bit better. So transformation is basically the process of taking in foreign DNA from the environment's. Transduction really has to do with a virus, right? Actually, they're called phages. Phages are viruses, but in this case, this is a bacteriophage we're dealing with because we're just talking about bacteria. So it's the movement of genes between bacteriophages. So you can see a phage right here, and phages have specific structures. However, they just basically dock onto the cell, and this is going to be a donor cell, okay? Even though it's receiving DNA from the phage, it's still going to donate DNA to other bacteria. Now, you can see the phage, it has its own genome. It's going to basically insert its genome into the donor cell. And here we could see the donor cell has a chromosome, and on the chromosome it has these two alleles. Okay, so it's positive for A and positive for B. Now, once the phage DNA gets in there, then it's going to start replicating and making more phages. Okay, so this donor cell is going to basically be the host for more phages to be produced. And when the phages are produced, they're basically going to be, part of the DNA from the donor cell, and of course the phage DNA is going to be incorporated into each of the phages. So here we could see one of these phages has that A allele. Right. So here, let's just say that phage, right, is released. Eventually, the cell's going to die because it's producing too many phage particles. It's going to die, and then this phage is going to go infect a recipient cell. So here it's going to insert that DNA, and here we're tracking that A allele, right? So the recipient cell is going to be similar in terms of genetic material to the donor cell. So there's going to be crossing over going on here. So we've got crossing over and exchanging of equal parts of DNA. So now this recipient cell is going to receive that piece of DNA. And here we can see that it's received that A allele. This can continue over and over and over again until many of the genes are going to be spread to a bunch of different recipient cells. So conjugation involves one of the structures that we discussed in a previous section of this chapter is the sex pilots. Okay, so you can see we have the sex pilots reaching from one bacterium to another. And in order to produce the sex pilots, which is basically a mating bridge, the organism must have what's called the F factor. So it's really a gene, probably a set of genes that are required for them to be able to produce this mating bridge. I'm going to look at a couple different ways in which conjugation can occur. And in this first way, this is where the F factor or F plasmid is present inside the donor cell. Okay, so the donor cell has a brown chromosome and the recipient cell has a green chromosome because they're considered, that's supposed to represent genetic differences, so they're not genetically identical. And here you can see the recipient cell does not have the F factor or the F plasmid. So basically the F plasmid is a piece of DNA, a small extra chromosomal piece of DNA with the F factor genes on it. So what's going to ultimately happen, and I basically skipped a couple of the pictures just to make it a little bit simpler, is that this F plasmid is going to be replicated. Okay, and the genes on it are being expressed because you can see that there's a mating bridge formed. So the donor cell is the one that actually made the mating bridge and then connected to a recipient cell because the donor cell has the F factor. Well, it's going to transfer that F factor to the recipient cell. So this is going to be replicated, and then the copy of that DNA is going to be transferred via the mating bridge so that the recipient cell now has the F plasmid. Right? So that ultimately shows the result. And they're still genetically different because the main chromosome is still different between each other. It's just the F plasmid was transferred between the donor and the recipient. Now, there could also be R plasmids, and those are ones that actually carry genes which allow antibiotic resistance. So those, you can see, can be transmitted from donor to recipient as long as also the donor does have that F factor. So again, the F factor is needed in order for the R plasmid to be transmitted. The way that this can happen is it's basically called high frequency conjugation or transformation. So what happens here is that there's a donor cell and a recipient cell. However, the F factor is incorporated into the genome here of the donor cell. And of course, the recipient cell does not have the F factor. And you can see a couple alleles highlighted. So the donor cell has the A and B allele. However, the recipient cell has the other allele, A minus, B minus for those particular genes. Now, what's going to happen is that this chromosome is going to be replicated, including the F factor. And again, the F factor is being or the genes are being expressed so that the donor cell can create that mating bridge and allow the transfer of that chromosome plus the F factor to the recipient cell. Now, because there are regions, they both had the same genes, right? So it's just different alleles. Because there are regions that are similar, there's going to be crossing over, and then now we have a recombinant bacterium. So here, what happened is that the A plus allele was incorporated into the genome of the recipient cell. Okay, so let's move into a little bit about metabolism. Now, there's other methods of metabolism, but we're going to talk about metabolism in terms of their ability to obtain carbon. How do they get their carbon? How do they make carbon? Okay, so prokaryotes can be divided up into categories based on how they obtain their energy and how they obtain their carbon and the different combinations of that. So in terms of energy, some prokaryotes can be what's called chemotrophic or chemotrophs. So chemotrophs obtain their energy from organic or inorganic compounds. Okay, so it could be organic or inorganic compounds or chemicals. Now, organic chemicals would be like organic acids, something, of course, that contains carbon. Inorganic things might be things that, like hydrogen sulfide, ammonia might be one of those things. Now, in terms of energy, some prokaryotes can be what's called phototrophic or phototroph. Of course, photo meaning light, right? So these organisms obtain their energy from light. Now, how do they get their carbon? Well, some of these kind of overlap, right? So those, the term for this is a heterotroph or heterotrophic organism. It obtains carbon from carbon compounds that are not within themselves, right? They can't make their own carbon compounds. Now, they could also be autotrophs. They obtain energy or carbon from organic compounds, however they make it themselves. This, right? So when we combine them, this gives us 4 major modes of nutrition. We've got the photoautotroph, obtains energy from light, and then carbon from usually carbon dioxide, and they make their own food, right? So they make their own carbon sources from gaseous sources. Chemoautotrophs are those organisms that obtain their energy from organic or inorganic compounds, and in this case, they're autotrophic, so they can actually make their own food. So basically, the carbon is going to come from something like carbon dioxide or bicarbonate. And then a photoheterotroph is going to be an organism that obtains their energy from light. And then, of course, being a heterotroph, they get their carbon from a external carbon containing compound, like organic compounds. And then, of course, a chemoheterotroph, which is what we are, right? So we obtain our energy from carbon containing compounds like organic compounds, and we obtain, of course, our carbon from that. So we must take in it externally. We ingest our carbon sources. All right, so those are the modes of nutrition, and this is kind of related to metabolism. Also worth it to mention a couple additional nutritional categories. Now, the first one applies to bacteria and archaea. The second one may apply more to organisms that we're going to discuss later, actually in the next chapter. But let's just kind of mention it now anyway. So there's those that are lithotrophic. Lithotrophic organisms use reduced inorganic materials for energy. Okay, so basically there's chemical reactions that take this reduced inorganic material or minerals, right? And then convert them to oxidized forms of that mineral. And this releases energy. So this is where they get that energy. Now, when we look at heterotrophs versus autotrophs of the litho type form of energy, we have lithoheterotrophs, which are just basically going to get the energy from the reduced inorganic minerals, but their organic molecules are going to, or the carbon is going to come from organic molecules. And then the lithoautotroph is going to, of course, use the reduced inorganic minerals for energy. However, carbon is obtained from carbon dioxide. Now, there's organisms that are mixotrophic, so they can actually convert between autotrophic and heterotrophic nutritional categories. One example is a euglena, which is a protozoa. Of course, that's going to be the subject of the next chapter, but it's worth it. Let's talk about the modes of actual metabolism. So metabolic modes. So in terms of the role of oxygen in metabolism, right? So you know that we require oxygen, but not all organisms require oxygen. In some cases, oxygen can be toxic. So again, in terms of prokaryotes, their metabolism with respect to oxygen can vary. Now, there's what's called an obligate aerobe. Okay, so obligate aerobes require oxygen. So it's obligated for them to be in the presence of oxygen. Obligate anaerobes, so anaerobic means no oxygen, right? So these are organisms, and prokaryotes specifically, that cannot be in the presence of oxygen. Okay, so if oxygen is present, often it can kill them. And then we have those that are facultative anaerobes. So they're anaerobic organisms. They can survive in the absence of oxygen, like I have here. However, they can also utilize oxygen, right? And a good example of that would be yeast. So yeast can ferment. So it undergoes fermentation, specifically alcohol fermentation. So this occurs in the absence of oxygen. So no oxygen doesn't kill them. And then the presence of oxygen, they utilize it. So they undergo aerobic respiration. In terms of nitrogen, some organisms can convert nitrogen gas into something usable. So you can't really use nitrogen gas to do something directly. It must be converted to ammonia. So if you look at this cycle right here, we've got nitrogen gas. Some organisms can convert nitrogen gas to ammonia. Okay, this is called nitrogen fixation. Here's an example. This is actually a cyanobacterium. It's called Anabaena. And there are two types of cells, right? So there's the photosynthetic cells, and there's what's called the heterocysts. The heterocysts are the cells that actually can fix nitrogen. So they can actually fix the nitrogen. They have the enzymes required to do that. And then the ammonia can be converted or transferred to the photosynthetic cells. And then the photosynthetic cells can fix carbon, right? And then transfer the carbon-containing compounds to the heterocysts. So they this is an example of cooperation, which we're going to talk about later. So that's nitrogen fixation. Some organisms take ammonia and then they convert it to nitrates and nitrites. Now, this is called nitrification. And then some can basically utilize the nitrates and nitrites typically found in soil and then convert it to nitrogen gas. So there's a cycle, right? So this goes back into the atmosphere. This is called denitrification. All of these are important, as well as those organisms that can utilize carbon and those that produce carbon dioxide for the atmosphere. Now, decomposers are organisms that can break down dead organic matter and basically use up wastes that are found in the atmosphere and in the soil. And some of the major decomposers are bacteria and then also fungi. So those are at the center of this right here. Now, you can see that we have these bacteria that are denitrifying. So remember, denitrification produces nitrogen gas, which goes back into the atmosphere. And remember, they utilize the nitrates and nitrites that nitrifying bacteria can produce. And the nitrification involves, remember, the conversion of ammonia into nitrates and nitrites, right? So we have some that actually produce the nitrites and then some that produce the nitrates. And then some can produce both. And then, of course, the ammonia often comes from those bacteria that can fix nitrogen from the nitrogen gas in the atmosphere. So you can see this is a cycle in which many organisms are involved and they're kind of dependent on one another. All right, so let's talk about, interesting habitats. So I kind of opened up with, in the previous segment, that prokaryotes are considered the masters of adaptation because a lot of them can adapt to their environments, and some of these environments are pretty extreme. So let's talk about some of these extreme environments. So the term for organisms that live in very extreme environments is extremophiles, right? So halophiles, they live in very, very highly saline environments. So this means very high concentrations of salt or sodium chloride. For example, the salt flats, right? So there could be some organisms that can survive that area. However, most organisms would not. There's thermophiles. Those live in examples would be a hot spring, right? So they live in a very, very hot environments. Now, there's different degrees of thermophiles. There are hyperthermophiles, which can survive in conditions above 120 degrees Celsius. There's acidophiles. They live in areas that have a very low pH, which is acidic, of course. And then we have the alkalophiles. Those are going to live in an area with a high pH. So it's more alkaline or basic. Again, these are extreme conditions, right? So most organisms cannot survive in these. Now we have piezophiles, often called barophiles. They live in areas where there's a very high pressure. So it's much greater than what we could handle. And then we have also ionizing radiation resistant organisms, or IRROs. Those organisms can survive ionizing radiation, which we would not be able to survive. So very, very high energy UV radiation and X-rays. Methanogens are quite interesting. They live in marshes and they produce methane, but they're strict anaerobes. So again, this is going to be something where this is an unusual condition where they cannot survive in the presence of any bit of oxygen. Now anaerobes are pretty common, but it is pretty extreme environment for. So let's talk about ecology. So this is more like maybe perhaps interactions with other organisms, right, and the environment. So I already mentioned this already. We have what's called metabolic cooperation. Cooperation can occur within a species and it can occur between species. This is an example of one that occurs within a species. So this is all the same species, but however, you can see the cells look different. And I already mentioned this, right? So this is an example of a cyanobacterium. And this is a bacterium in which some of the cells, both the types of cells, they cooperate with each other. So we have those that are photosynthetic and those that can fix nitrogen. And then they're going to exchange these metabolic processes in a cooperative fashion. Have those that can happen between species. A really key example would be a biofilm, right? So biofilms basically starts with a bacterium that is like a scout, right? So it finds an area and it attaches to something. And then it starts to grow a colony and produce a matrix. And this matrix is like a it's like a house, right? So it can serve as like a house so that other species can enter that house, and then it's like a party in there now, or a community, I guess you can say. But they basically attract other ones to come. So they come, and then this grows, and then it becomes a mature biofilm. And then eventually parts of it will detach and then again complete the cycle, right? So this will continue elsewhere. So there'll be another area in which the scout bacterium attaches and then starts a new biofilm. One example would be, this is of course a drastic example of plaque, right? Plaque is an example of a biofilm. Now, biofilms are quite interesting in that when organisms belong in a biofilm, they're kind of cooperative, so they're working with each other. It takes 100 to 1,000 times more antibiotic power to be able to treat them because they're quick. So another one would be what's called symbiosis. So you've been introduced to, of course, you should be familiar with endosymbiosis, but symbiosis occurs between organisms where there's not one inside another one. So there's generally going to be a larger host and a smaller organism called the symbiont. So the symbiont kind of lives on, sometimes in parts of the host. One example would be the root nodules, right? So this is rhizobium. It's a bacteria that lives in the roots of plants. So we've got the plant, which is the host, or the plant root, which is the host, and then nodules that are formed at the plant root underground. Often the rhizobium can fix nitrogen so that it can fix nitrogen, converts it to ammonia, and then the ammonia can be used by the plant, right? So that provides a benefit for the plant. And then, of course, the plant provides a home for the rhizobium. Commensalism is where one organism benefits and the other one isn't harmed or really has no benefit. So sweat pores would be an example. So basically, there's bacteria that kind of live in your sweat pores. This might sound a little bit gross, but they actually live in there. So you're providing a home for them. Now, it's not generally not, it's a benefit for them. It's generally not harmful to you unless they do overgrow. There's what's called parasitism. Okay, so one organism is called the parasite and generally harms but doesn't kill the host in most cases. Parasites that cause disease are called pathogens. So something can be parasitic, however, not considered a pathogen, right? So here we have a couple examples, right? So we have Lyme disease. It's caused by a bacteria, which is often they're transmitted between other species, right? So we are basically the definitive host or the final host, but the ticks or often mosquitoes are the vector. Now, a lot of them cause disease by releasing toxins, and there are two types of toxins. There's what's called exotoxins and endotoxins. Exotoxins are released by living cells. So if you can imagine from General Biology 1, remember you've got these vesicles that are going to release contents of them. So that's the same idea. It's a living cell that's actually releasing this, usually an enzyme or protein or some other chemical that causes the damage to the host. Now, there could also be endotoxins as well. So endotoxins result from the breakdown of the bacterium or the parasite or the pathogen in this case, right? It starts to break down and the components of that pathogen cause disease or damage to the host, okay? Usually the host is trying to react to it with their immune system and that causes even more damage, right? So there's two types of toxins. We have the exotoxins and the endotoxins. So mutualism is a type of symbiotic relationship where the the organisms that are involved in the relationship benefit. So in humans, we have mutualistic bacteria. So we have bacteria that kind of live in us, and they get a food source as they're digesting food that our intestines may not be able to digest. And this, of course, is going to help our gut health, really, and then, of course, assist them in obtaining nutrients as well. Now we have, this is another example, but there's a ton of different examples for mutualism. This is a really cool one, though. We've got this fish, which have these glowing bacteria that live on the fish, and they obtain nutrients from the fish. And the fish obtain the benefit of having this light, which can attract prey, and then, of course, maybe attract mates as well, right? So maybe the females of this fish are more attracted to a brighter, bigger, brighter flashlight, I guess, if you will. Remember, we've talked about this before with the intersection. So let's talk about some practical uses of bacteria. Okay, so practically speaking, bacteria are prokaryotes in general used in research and technology. So we often engineer bacteria to produce things that we're studying. In fact, I've done that before, right? So I've engineered E. coli and yeast to produce a gene that I was studying, or actually produce the protein, and then I took the protein out of that bacteria and then was able to study it that way. Now, E. coli are often used for this. That's basically the process of this is called gene cloning. And then you've probably heard of the CRISPR system, CRISPR-Cas system, which can be used for gene editing, right? So this can be used to potentially, in gene therapy, to potentially cure diseases like HIV. Bacteria can also be used to make plastics and also break down plastics. We'll see that later on. But some bacteria can synthesize plastic, and then it can be stored inside that cell, and then you can kind of extract it later. Some bacteria can be engineered to produce ethanol. And this allows us to be able to use the bacteria to use up any waste that we might have, like biomass and then some corn waste and switchgrass. The process, or this thought of what's called bioremediation, is the use of organisms to remove pollutants, like an oil spill, right? So some bacteria can metabolize different types of oils, which might be dangerous for other organisms. And as I mentioned before, we have those bacteria that can actually break down plastics. Here's that example right here. So this is a bacteria that can actually eat plastic. It can metabolize plastic. So this can help the environment. So the main point of bioremediation is to basically clean up the environment using bacteria to do it. Now, in summary, we talked about several things about prokaryotes and the way they live, right? So their lifestyles, including how they reproduce, different modes of reproduction, and how they obtain their carbon and energy, different types of extreme habitats that some bacteria can, or prokaryotes can survive in, and their ecological interactions, and of course practical uses of bacteria. So the next segment's going to be on the groups of bacteria, if you do these in order.